All right, hi guys. So this week we're talking about genetic engineering and gene cloning. And a lot of people struggle with this one. So I figured I would do it kind of in a lecture format. Make a little video so that I could familiarize you with the sometimes difficult content here that can be difficult to extract out of the readings. A lot of you are finding that you're struggling to get it all. So I'm just going to run over it and see if hopefully if I explain it in certain ways, it can help you guys understand it in a way that the readings couldn't do. All right, so far, oh boy, why isn't this working? Okay, so far we've been talking about eukaryotes. Now, what is a eukaryote? A eukaryote is a cell with a nucleus. Every single cell we've been talking about has a nucleus, remember? And that's where the DNA was. And remember, their DNA was linear. What I mean by that is, so we've got a eukaryote that's got a nucleus. Their DNA is linear, meaning that it's in a line. You see, here's a karyotype from a human. It's linear. You've got one end here and another end here. It's in a straight line. Now, bacteria don't have a nucleus, and they don't have linear DNA. So that means they can't be eukaryotes. So what are they? Bacteria are what we call prokaryotes. Prokaryotes are different from eukaryotes in that they don't have a nucleus, so they're just normal, and the DNA is kept in the cytoplasm because since there isn't a nucleus to keep the DNA in, we have to keep it somewhere, and so it just floats around on the inside of the cell. Now, here's the interesting thing about that, though, because remember we told you that on the eukaryotes, they had linear DNA. That means the DNA was in a line, like this green line is. Well, part of the reason why eukaryotes have to keep their DNA in a nucleus is because there are enzymes that called exonucleases that look for the ends of DNA, and they chew on it like Pac-Man. And they will chomp all the way through until the DNA is completely annihilated. If you're a bacteria and you don't, now that's fine for a eukaryote, but if you're a bacteria and you don't have a nucleus to protect your DNA is going to get destroyed. That's bad. We don't want that. So we need to come up with our own way to protect our DNA, and that answer is making our DNA a circle. See, if we make our DNA a circle, there's no ends anywhere, and I can throw as much exonuclease in there as I want, and the Pac-Man will never find the end of the circle. And so my DNA is good. It won't get destroyed. That's why bacterial chromosomes are in circles so that they won't get annihilated. So we find that they've got these circles of DNA sitting in the cytoplasm. Now, what is a plasmid? We've been talking about it kind of in the readings, and a lot of people are very confused about what one is and why it isn't a chromosome. A plasmid is a circular loop of DNA, just like the chromosome is. It is DNA loop, but it is not a chromosome. Now, here's a couple of differences between the two. First off, chromosomes and plasmids, even though they are both made out of double-stranded DNA in a loop, the plasmids are tiny compared to the chromosomes. The chromosomes carry a ton of genes, whereas the plasmids carry only a few. The additional thing is you can have lots of plasmids, and you can have lots of plasmids of various sizes too. They don't all have to be the same. And the interesting thing too is these things, although they carry genes, chromosomes carry what we call essential genes, whereas plasmids carry non-essential genes. Now, what do we mean by that? Well, let me give you a bit of an analogy here. Building a house. We've been telling you this entire time that DNA is the blueprint for a cell. Well, a blueprint gives you instructions on how to build a house. It gives you the structure. You know, a house is one of the essential things that a human needs, shelter. And so it needs to keep out the elements. It needs to keep us warm. That's great. And the blueprint carries the essential information on how to build the house. Now, we can build the house and be just fine. You don't need furniture. You don't need anything like that. Chromosome is like the house plans. A plasmid has the interior decorating instructions. So whereas the chromosome is like the house plans, the plasmid is kind of like Better Homes and Gardens magazine, where it gives you instructions on how to make your house beautiful on the inside and make your life nice and happy while you're there. It improves your quality of life. It's not necessary to have those things. You don't need couches. You don't need lamps. You don't need rugs. But it's nice to have because it makes your life nicer. It improves your quality of life. Non-essential, but nice to have. Where the chromosomal DNA gave you the structure of the house, Better Homes and Gardens can give you a really nice living room. I'm jealous of this living room. It's a very nice living room. So these plasmids are interesting because bacteria can actually pass them back and forth. And there's a couple of ways that they can do that. You can either pass it from bacteria to another bacteria. You can actually pass it from a virus to a bacteria. Or they can simply pick up a plasmid from their environment. This is actually very interesting. People have asked why plasmids would be in the environment. Well, when bacteria die, they explode. And if they explode, then that means the plasmids that were in their nucleus are now just floating around free in the outside world. And these guys can then pick them up. What's interesting is this is actually implicated. This is the reason why they have you take your full dose for antibiotics. And we can, I'll cover it now. If one bacteria has a gene which is non-essential, such as antibiotic resistance. See, you don't need antibiotic resistance in order to constitute a bacteria. You know, the essential genes are like the ones that do DNA replication, the ones that build the cell wall, the ones that help me move, the ones that help me eat, things like that. But I don't necessarily need to be antibiotic resistant to be a bacteria, but it is nice because it helps me survive. So if you don't take your full antibiotic run, then it doesn't kill all the bacteria. And let's say one or two of the bacteria in your body have a gene on a plasmid for antibiotic resistance, a gene that makes an enzyme that can chop whatever antibiotic you're using in half, and it no longer works. Well, if you don't take the entire stretch, if you do take the entire stretch of antibiotic, then it kills everyone but the guys with the antibiotic resistance, and there's so few of them that the immune system can go take them out. But if you don't take your entire course, then that leaves a few of the people that don't have it alive, and then the bacteria play a Lehigh tree of life scenario and copy their plasmid and pass copies to all their friends, and then when you go back to try and use that medication again, it's not going to work because everyone's got the plasmid. That's why it's important to take your entire course. Let's see here. Okay, so so now, how do we use this? We understand what a plasmid is. Why are we learning about it? Why do we need to learn about it? Well, we can hijack this system in order to use bacteria for our benefits. Let me go back to my Better Homes and Gardens example here. Let's say I am a campaign manager for a gentleman, and I want people to vote for this gentleman, Andrew Richter. I want people to vote for him. Well, I could put in an ad. I could go to Better Homes and Gardens magazine and insert an ad in there somewhere. Everyone who then picks up the magazine will see my ad, and I can cause people to, in addition to making their house nice, put up my signs put up the little campaign signs on their front yard. That's what I want them to do. So by putting in the instructions, in addition to what they're actually there for, which is to make their house pretty, I inserted my little gene, my little vote for Andrew Richter, and in addition to making their house pretty, they also will put a sign out in front of their house. I got them to do what I wanted to by inserting a little piece into their magazine. And now they can pass that, they can photocopy their magazine, give it to all of their friends, or give subscriptions to their friends, and now all their friends will see it too, and they will make it. So far, so good? Okay, here's a good example. Insulin. Insulin is necessary for diabetics to survive. A lot of diabetics need insulin or can't make enough insulin. Their pancreas doesn't work. And so insulin is essential to get sugar inside your cells. So without insulin, you can't get sugar into the cells and the cells will die, and that's why they feel faint. Well, insulin, the only way you can get it naturally is from human donors. You have to basically go to the plasma donation center, get a bunch of plasma, filter it down until you can get insulin out of it, and then give that to the patients. That's expensive. Not only do you have to pay the donors, but then you have to siphon it down and filter out all the other stuff that you don't want and just get it down to pure insulin. And diabetics need a lot of this stuff. So we need to be able to make this cheaper. This is where we bring in plasmids. Plasmids carry genes on them. Well, what if I can take an insulin gene from a normal human DNA? I get a sample cell from someone who has an insulin gene on them that works normally. They're able to make insulin fine. And I want to move it over so that I can put it into my bacteria and turn them into a factory. Because watch this. If I take this insulin gene here, put it on a plasmid, then put that plasmid inside a bacteria, then that bacteria will turn from being just a normal bacteria into an insulin factory for me. Because they're going to uptake any plasmid that I give them, and then they're going to make whatever genes are on said plasmid, including my insulin one. So I can actually have bacteria manufacture insulin for me, and then I can make a big pot of these bacteria that are manufacturing insulin. And then I can siphon off the insulin off the top of the pot, and they'll make gallons of it for me every day. And then I can siphon that up, bottle it up, and sell it for way cheaper than I could have gotten it earlier. And now suddenly being diabetic isn't such a monetary strain as it used to be, because I don't have to get it from humans. I can get it from bacteria. It's the same substance. There's no difference, but the bacteria made it, and I don't have to pay a bacteria to work for me. So then I can sell it, make money. The diabetic people are happy. Everyone's happy. Now, the question about this week is, how do we do that? Because we went up here, and I had to get that insulin gene onto the plasmid in order to put it into the bacteria so that I could turn it into a factory and make insulin for me so that I could make money and make all the diabetics happy. So how do we do that? Well, we need to move that gene over. First step, we need to cut it out using scissors. Because notice, before it was in one big long line, but I don't want to insert the entire chromosome in there. That would make a plasmid that's too big. I don't want the whole chromosome. I just need the insulin gene. So I need to cut out the insulin gene using scissors. And these scissors, they're molecular scissors, I suppose. We call them restriction enzymes. Restriction enzymes are enzymes that are capable of cutting across double-stranded DNA in particular spots. So we use these molecular scissors, restriction enzymes, cut on both sides of the insulin gene. And then we also have to cut the plasmid, because notice it's a circle. So in order to insert my gene in there, I have to cut the plasmid using scissors. And then I can insert my insulin gene into the plasmid. Now these molecular scissors, they can come in here. These molecular scissors need to look for a specific spot. You might think of it like the dotted line. You always cut at the dotted line. Well, these scissors look for a particular sequence. And we call those palindromic sequence. And each restriction enzyme has its own. So what is a palindrome? A palindrome is any word that or phrase that can be written or that is spelled the same way forwards and backwards. Like race car is a classic example. R-A-C-E-C-A-R, spelled the same way in both directions. There's numerous ones of these, but that's the theory. With genetics, a palindrome is where going from the two parallel strands, the anti-parallel strands, are spelled the same way going 5' to 3' on both strands. Like this. Here's an example. A-G-T-C-G-A-C-T going 5' to 3'. Well, A pairs to T, G pairs to C, T pairs to A, C pairs to G, G pairs to C, A pairs to T, C pairs to G, T pairs to A. Everything's dandy, but look at this. If you go 5' to 3' this direction, it's A-G-T-C-G-A-C-T. A-G-T-C-G-A-C-T. It's palindromic. Notice they're complementary to each other, but 5' to 3', 5' to 3', they're spelled the same. So let's practice how to make these things, because they actually have you do this on the homework. And every single person I've talked to has messed up the first like 20 times they've tried to do it, myself included. So let me pause my video here. Okay. There are 3 ways you can go about this, and I'll show you all 3 of them. I'll show you the one that I use. But it may be better to start practicing the other 2. And I don't know. I show you all 3 so that you can pick whichever one you favor and use that one. Because different people think differently. So I'm going to show you 3 different methods, and you can pick whichever one you like the most. The first method is write out the first half of one of the strands. So let me go, I don't know, A, C, G, A, C, G. I'm making a 6. Base pair one, because I can't give you the answers for your homework. ACG. I wrote out half of one of the strands. Now write the same bases on the opposite end of the complementary strand. So notice, I know that if I'm going to do A, C, and G, then in order for it to be palindromic, this one has to be an A, that one has to be a C, and that one has to be a G, right? Because it has to read the same 5' to 3'. Well, now, if I just go in there and do complementary base pairing, C pairs to G, G pairs to C, T pairs to A, G pairs to C, C pairs to G, A pairs to T, ACG, CGT, ACG, CGT, it's palindromic. Here's the next method. Write out the first half of one of the strands. A, C, let's go T this time. ACT. Okay. Step 2, write the complementary bases in reverse order on the same strand. What we mean by that is look at the T. T pairs to A. A is going to go right here. Now go the next one. C. Almost think of it like a mirror image, but the complementary. So T pairs to A, C pairs to G, A pairs to T, and then if I go in here and pair it up, let me go this way. T, G, A, T, C. I go this way, A, C, T, A, G, T, A, C, T, A, G, T. It's palindromic. Here's the third method. Let's go TGC. Oops, I should probably have that. TGC. Okay. Write out the first half of one strand. We just did that. Write in the complementary bases to make complete base pairs. So here I'm going to go A, C, G. I've made half of my calendar this way. So the G will go here, the C will go here, the A will go here, the C will go here, the G will go here, and the T will go over here. TGC, GCA, TGC, GCA. It's palindromic. You can pick any of those three ways. Those are just the ways I go about it. And it just depends on whichever one works best for you. So yeah, I would definitely practice making them, though. They're not as hard as they look initially, especially once you get down these rules. You can make them out of any combo of letters, and you can make them however long you want. The rules work with any length of palindrome. Oh, shoot. You couldn't see my screen that whole time. Okay, let's try this again, where you can actually see the pictures now. I am so sorry about that. Okay, 5' to 3'. Let's go A, C, G. So G, so write out the first half of one strand, A, C, G. Write the same bases on the opposite end of the strand, complementary. Go in the opposite direction. So A has to go next to the 5', C then goes there, and G has to go there. Then I just fill in the remaining bases as they pair. C, G, T. And T, G, C, it's palindromic. ACG, CGT, ACG, CGT, it's palindromic. Okay. Write out the first half of one strand. GCT. Dang it. It's been one of those days, guys. GCT. Okay. Write the complementary bases in reverse order on the same strand. Dang it. I am just having a rough day, guys. I am so sorry. I just did that up here. So let's do method 1 down here. Okay. Oh, no, just kidding. Okay, this is method 2. Man, my brain is so dead here. Okay. Complementary bases in reverse order on the same strand. This is what we mean. T, what does T pair with? A. So A will actually go next to T. Think of it like a mirror. What pairs to C? G, so G goes here. What pairs to G? C, so C goes here. Then you do the complementaries. C, G, A, T, C, G. 5' to 3', it's GCT, AGC, GCT, AGC, it's palindromic. Here's the third method. A, T, T. Well, what pairs to A? T. What pairs to T? A. What pairs to T? A. So I wrote that out. Now make the mirror image and flip it. So this is kind of what we were doing before. But A goes there, A. A goes there, A. And T goes there. And then you pair them up. T, T, A. A, T, T, A, A, T. A, T, T, A, A, T. It's palindromic. I'm so sorry about the you not seeing the first part there. Okay, we're back. All right, so this is a sequence given to you in the homework. They're telling you that this is a beta hemoglobin gene. It's the same story as my insulin problems we were doing earlier. I'm just doing beta hemoglobin instead of insulin. So I need to be able to cut out this gene and put it in a plasmid so that I can stick it in a bacteria and turn them into a beta hemoglobin factory for me. In order to do that, I want to cut before here, and I want to cut after it, like there and there. Now, why do I want to do that? Because if the gray area is the gene, why would I want to cut in the white? Well, think about it. If you cut in the gene, it's going to break the gene. Remember, if you chop a gene in half, it's not going to work. Think of it like this. You know how when you go to primary, you've got a lot of these, or they have the kids, you know, when Christmas or Easter or anything like that comes around, they give them like a little coloring sheet like this, and then you have to cut it out and paste it onto a stable and make the Christmas scene. Well, let's say we're trying to cut out Mary and Joseph here. If the little kid goes in there, colors it in, and then cuts right there, they cut on the red lines. Is that what we want them to do? No, because I mean we're missing the entire top half of Joseph. We end up only sending the top half or the bottom half of Joseph and the bottom half of Mary to the manger scene, and you end up with some really creepy Tim Burton Christmas where only the legs of Joseph and Mary show up to the manger, and it's like, wah! And give child nightmares for the rest of their life. Don't Tim Burton your jeans, okay? Don't chop in the middle of a gene. Whatever you do, don't chop Joseph and Mary in half. Don't Tim Burton your jeans. We don't want just the legs showing up. That's creepy. We want all of Joseph and Mary. That's why we have to cut before and after. We want to go all the way around Joseph and Mary so that all of them shows up. Now, that's why we need to cut in those white spaces. Another interesting point, though, let me go back here. Notice, on this picture, we said we wanted them to cut there, but what if they cut out here? Well, is that a problem? No, it just means it's bigger. It doesn't matter how much white paper I get around the outside of Joseph and Mary, as long as I get white paper around the outside of Joseph and Mary. So what that means is on my sequence here, I could cut it right here next to the gray, or I could cut it clear the blazes out here, or I could cut it over here, or I could cut it, and then down here, I could cut it here, I could cut it over here, I could cut it over here, I could cut over here. Doesn't matter how much white space I get, as long as I get the gray and I don't cut it. So I have to cut before and I have to cut after, and it can be anywhere in there. The white space doesn't matter. We can have some of that. Okay. So these enzymes, the restriction enzymes, the scissors, we've got this list down here on the left side, and we have their restriction sequences here. Now, these enzymes are interesting because they look for a particular palindromic sequence, but why palindrome? Well, it cuts between the same two bases on both strands. Notice it's between the G and the G here, the G and G there. C and C here, the C and C there, and so on. It cuts between the same bases on both strands. Well, that means that sometimes, here we go. So that means that when you cut it, you end up with a zigzag pattern here, where it cuts through the sugar phosphate backbone, comes across the hydrogen bonds in the middle, and then back down again. That gives you almost like a puzzle piece effect with 4 bases hanging out. We call that sticky ends. The reason for that is hydrogen bonds, these guys are now free hanging in the water. They don't want to be free hanging. They're hydrophobic, so they're going to want to pair up with a complementary strand, another GATC, just as fast as humanly possible. That becomes important later. Down here, notice on the SMAI, we don't want to use something like that. What's wrong with SMAI? Well, notice it doesn't cut in a step pattern. It cuts straight through. We don't want that because, notice, if we come in here and chop it like that, sorry, you can't see past my X. If you chop it like that, then you don't end up with any hanging over. There's no sticky ends. That's going to become really important later because our sticky ends are important for getting the DNA to stick back together again. So we don't want to use something like SMAI, even if we can, because it will shear off my ends. Now the DNA won't stick back together again, and I've just ruined the whole gig. So we don't want to use ones that cleave straight through. We want them to cut in a zigzag pattern so that it gives me sticky ends. We want to cut it like that. Okay. So we have to come in here and cut it with the scissors. We need to cut the DNA, cut out my gene, and we also need to cut the plasmid. Because notice, remember before the plasmid was a full circle, we needed to stick the plasmid using the same scissors. So here's your plasmid. So we need, here, what this is saying is this is 5,500 base pairs long. If we tried to write that out base pair for base pair, it would take pages, plus it would be a waste of your time trying to find all of the restriction sites for each of the enzymes. So what we did was we told you that BAM-HI has a restriction site right there. So if I throw BAM-HI in here, it's going to cut right there. Does that make sense? It's going to cut over here. Notice some of them have two, some of them only have, some of them have two sites, some of them only have one, and one or two of them only, or don't have any. That just simply means that there's only one of their restriction site in the DNA pattern, or in the DNA sequence on this particular plasmid, or there isn't, or you can't find their restriction sequence on there. So if you threw an enzyme in there that wasn't, if you throw an enzyme in here that doesn't have a restriction site on the circle, then the enzyme will just sit there and not cut anything because it won't find its own dotted line to cut. So it won't do anything. You just wasted a lot of money. So you need to make sure that whatever enzyme you pick has to be able to cut both the plasmid and the DNA. So we have to cut the DNA and the plasmid using the same set of scissors. Now here's why. It's all about the sticky ends. Notice the hangover here, GATCC, that's going to want to pair, or sorry, the GATC is going to want to pair with a hangover GATC here, right? It's not going to want to pair to a GTAC. So if I threw in NCOI and had it cut my plasmid, and then threw BAM-HI in to cut the DNA, I'd get sticky ends, but the sticky ends wouldn't match up. It would be like trying to stick two puzzle pieces together, and one of them has a square peg and the other one has a round hole. You're not going to get the two to stick together, and I need them to. So you might think of it like these fancy scissors, you know, the ones that cut in patterns. You know, you have a zigzag here, and some of them have a wave, and some of them have points, and etc. But if you try to cut one with wave scissors and one with a zigzag scissor, they're not going to match up. So you need to cut with the same pair of scissors on both of them, meaning you have to use the same enzyme to cut both your plasmid and both cuts on the DNA. It has to be the same pair of scissors doing all three cuts. So you have to pick which of these 6 enzymes is the only one of the 6 that can do all 3 cuts properly. Okay. Okay, so we find the sticky ends that match up properly. We cut the DNA out over here. We cut the gene out over here. We cut the DNA on the plasmid. Opens that up there. Now all we have to do is transfer the DNA over, use DNA ligase, if you remember, to seal the nicks in the backbone. And ta-da, then all we have to do is throw that in a test tube with a bunch of bacteria, and then subject those poor bacteria to cold, then hot, then cold again. And then they're stressed out, so they're going to stress eat. Because, I mean, if you ran if someone took you and threw you into a freezer for 2 minutes and then pulled you out of the freezer and threw you into a fire for 2 minutes and then pulled you out of a fire and threw you into the freezer again for another 2 minutes, you'd be stressed out too. So they basically stress eat. They force these bacteria to stress eat and pick up the DNA. That's the plasmids that I threw into that test tube, the plasmids that I made. So that means that I will get bacteria, because I caused them to stress eat, they're going to open up, make their membrane more permeable to allow the DNA to come in. now they've picked up my plasmid. See right there. So this is their chromosome over here. Now they've picked up my plasmid. But not every bacteria is going to. I'm going to get some bacteria that's going to pick up my plasmid and some that aren't, even if I subject them to the same treatment of the hot, cold, hot, or the cold, hot, cold. Sorry. So not every bacteria is going to take up the plasmid. So how do I tell who's going to take it up and who's not? Or who did take it up and who didn't? Because I don't want to invest money growing bacteria in my pot that aren't going to be making insulin or beta hemoglobin for me. So how do I tell the difference between a bacteria that took up the plasmid and one that didn't? Because I can't visually tell the difference. There's two ways we can go about this, two major ways. First off, so for the first one, let me take you back to my house example and kind of make it a bit more radical. Maybe this will help it stick in your mind because it's going to be really, really extreme. But so we have a house. We told you that the Better Homes and Gardens magazine made or decorated the house, and I put in my little ad in the newspaper to have people, or in the magazine to have people put my sign out front. Well, what if I want to be able to tell who put my sign out front or not? And I'm a really weird person that should never be voted for. Just bear with me for a sec. I come up with weird analogies sometimes. Well, in addition to putting in my ad that's going to cause them to put my sign out there, what if I put a second ad in that teaches them how to put a force field around their house? Now, that means that anyone that, let's assume the people are all just robotic, sort of, and they decorate their house with everything, and they put my vote for Andrew Richter on the outside, and they build their force field around their house. That means anyone that took the magazine is going to build a force field around their house. Now, that still doesn't help me at all. So I've got a force field around the house. That doesn't help me until I nuke the city. If I go in there and drop a bomb, it's going to level everything, except my house with the force field. So what I did was I prepared everyone who took my magazine and put my sign out front by giving them protection, and then I put them through a treatment which killed everyone except the people that took my magazine, which are perfectly fine and happy inside their force fields. It's kind of a dramatic example, and I'd hope you'd never vote for someone that would nuke a city to figure out who's on their team and who's not, but just bear with me. So here's how we do this in real life. Penicillin is an antibacterial. It kills bacteria. But some bacteria have developed a gene that makes an enzyme that chops penicillin in half. And it's on a plasmid. So what if, in addition to putting my insulin gene on the plasmid, I also put a penicillin resistance gene? I put on a gene purposely that makes the bacteria so that they don't get killed by penicillin. Then I stick it in the bacteria again, and I grow them on petri dishes, but the petri dish is going to have penicillin on it. So who are the only ones that are going to survive and grow to become these little bacterial cultures? The ones that took my plasmid and are invincible to my penicillin nuclear bomb. So if these little pink dots are my penicillins, my house is perfectly fine because I can drop as many penicillin bombs on this house as I want, and it's got the penicillin resistance gene because it took my magazine. Whereas everyone else is going to get leveled. So that means I end up with a petri dish that has just the bacteria on it that ate my plasmid. And then I can go take those and grow those in a pot, and poof, I can make lots of insulin. But by making them antibiotic resistant, in addition to, or by putting antibiotic resistance on the same plasmid as my insulin gene, I can tell which bacteria are going to make insulin for me and kill everyone else that's not going to. The other one is green fluorescent protein. This is the other method. Green fluorescent protein is taken from a jellyfish, and it glows green, hence the name, green fluorescent protein. Well, we scientists have found the gene for green fluorescent protein. So what if instead of putting penicillin resistance on there, I can put on a gene for green fluorescent protein? Now, how can I use that to tell which bacteria took up my plasmid or not? Well, any bacteria that took up my plasmid is going to glow in the dark. And anyone that didn't isn't going to glow in the dark. And then I can just sit there and pick out the ones that glow in the dark and leave behind everyone else, and then poof, I can make insulin in a pot that glows green, and everyone thinks they're from a nuclear test site. But you follow me. In fact, they've got several different varieties of these green fluorescent proteins that they've modified to get different colors. So here's a picture that you saw in one of your videos that she just barely flashed by. But this is a picture that had from a scientist that had way too much time on his hands. And he made several different strains of bacteria. A strain is just a species, basically. It's a subspecies, kind of like blonde hair or brown hair. He made several different strains of bacteria that have different colors of green fluorescent protein in there. So the original green fluorescent protein fluoresces green like this, and then they've got various bacteria with different colors. And then this scientist sat down and painted a picture and then grew it for a couple of days, and poof, he's got a painting. And I just think that's really fun. Way too nerdy, and seems like something I would do, but it legit glows in the dark like that. So that's it for the video. I sincerely hope that this helps clarify some confusion. If not, please email your teacher or email me. We'd be happy to help. And yeah. Oh, I think the one other thing, transformation. Transformation is the process of moving a gene, transformation is the process of bacteria picking up the plasmid from outside their environment. That's transformation. So that's what we're doing with that whole hot, cold, hot thing. Alrighty. Well, thanks for watching. Thanks for putting up with all of my mayhem, and have a great one.